So good morning, everybody. Welcome to UAB Pathology Grand Rounds. This morning, we get to hear from one of our own, Dr. Melkani. Dr. Melkani originally received his PhD in India before coming to the US, where he completed his postdoctoral training in San Diego at Cal State San Marcos. He stayed on there. He liked it so much to join the faculty, where he quickly rose through the ranks to obtaining his tenure. And then he is one of our newest faculty members here in MCP, having joined us just last October, a few months ago. So we've been very fortunate to acquire him. And as you can see from his slide title, he's very interested in circadian rhythm. You can see the swirling clock and cardiometabolic disorders, but he takes a very unique approach. He uses the fly or Drosophila model to study these metabolism, cardiac diseases, and we'll see how he does that with the fly today, as well as a number of other genetic factors, taking full advantage of the long history of all the tools that are available to the fly. He's published 40 manuscripts in some of those slouch journals like Science and Nature Communications, those ones that, they take anything at. No, very wonderful publication record. I think he has the holy trinity when you consider the sister journals of Nature Communications and Aging Cell. But probably most impressively, he currently holds 3 NIH R01s. One is contact PI and 2 multi-PI, all studying various aspects of cardiovascular disease, Alzheimer's disease, and circadian rhythm. As might be expected with somebody with that impressive publication and funding record, he's also deeply committed to the training of the next generation, including undergraduate students, graduate students, both in the classroom as well as in the lab. He's very active and has been both in his last institute as well as hitting the ground running here at UAB, staying active in different committees, including grant review for the NIH. Without further ado, please join me in welcoming Dr. Melkani as he tells us about modeling and delineating the pathophysiological basis of cardiometabolic disorders. Good morning, Dr. Melkani.
Thank you very much for wonderful introduction. And I'm really pleased to be here. And one of the main reasons that motivate me from the beginning, first day I talked to Dr. Patel, the kind of collaboration. And I think we're gonna have more and more based upon, last 3-4 month conversation I have. But nonetheless, let me talk about the title already. Basically how we modeling different kind of disease. My primarily focus has been cardiac disease, some muscle disease, and recently start working on some neurological aspect, but all that how the circadian clock work on different tissue. So that has been the main motivation in the lab for last 5-6 years, especially since my independence. And for mostly, we use the Drosophila model. We have some human study also, including muscle biopsy, all these kind of things. But my primarily one is Drosophila. Why I use Drosophila model? That's the one of the, some basic idea why I want to give to because most of the people, particularly in the department, they may be not familiar with this model. So what it has is, it has at least 75% of human disease homologs. Okay. And then other advantages, it has short life. You can see from, this embryo to adult, you can get that in 10 days. And they live only for like 70 days. Not they have short life, but whatever those pathway, that uh, you see in human aging or any like the cardiac aging, you can see these kind of change in fly model. And then other thing also, what we can see if we are modeling a human, dilated cardiomyopathy versus SCM, so we can model that in the fly model also. So of course, it exhibit like, you can record sleep, circadian activity, memory, all these kind of experiment you can do. And other really huge advantage is you can manipulate, use your genetics and manipulate gene and see that, loss of function, gain of function, all these kind of things you can do really quickly. The best part is lot of mutant like RNAi, over 95% of the fly genome RNAi. Most of you know fly genome has been sequenced several years back. So we have about 95% of the fly genome RNAi line are available. And recently, lot of CRISPR line are also available. About 20 to 30% of the fly genome now CRISPR stock are also available. So it's a great model for doing high throughput screening purpose. Okay, I want to also give what kind of thing we can do. One idea called forward genetics. So we make a mutation. You can, add random mutation. There are multiple ways you can introduce mutation, and most of those mutation are available. So what you can do, you can study human congenital disease. Like I'm talking to a couple of clinicians. They said we have, some cardiac congenital disease. So that's also will be a great model, and we can use a reverse genetics, so we can identify where these mutation are, and then that's how we can test our hypothesis and drive the pathway. At the end, we can use those for like a drug screening purpose. I will talk a little bit later. The other idea is, if you look at in last 5-10 years, all those RNA sequencing, transcriptomics is really available. Anyone can do these kind of things. So what you can do is, after that, what is next step? So what we can do, we can just look at those disease variant and then look at the Drosophila ortholog, and using the reverse genetics, we can identify what are the target. Okay, if it is down-regulated, what it means at physiological level. So we can do all these kind of things in the fly model. Also, please, if I didn't mention, if you have any question in the middle, please feel free to stop me or ask me anytime. I really like that kind of one-to-one conversation. Okay, as I said, I primarily use Drosophila heart model. So if you want to look at the early cardiac development between Drosophila heart and human heart, I mean, not human, mammalian heart, I must say, are quite similar. Okay. Later on, they differentiate. Then you will have a chamber-like structure, all these one. The other important aspect is, if you look at those transcription factor, like most of the vertebrate transcription factor, they are present in the fly model also. And more importantly, if you look at the percentage of coding versus non-coding sequence, across those eukaryotic, you can compare those, fly and human. So having said that, all that means it's a great model for, doing some cardiac study. So my lab also, what we do is, this is really, I want to go a little bit on the adult heart. So unlike vertebrate heart, it's a long tube, okay? It has the valve and it has the ostrea. So valve is like that, same way we have a human heart that separates the chamber, same way the valve does here. The ostrea, it controls the flow of the hemolymph. When I say hemolymph, I'm talking about Drosophila blood. It does the exactly same thing except oxygenation. For oxygenation, they have a tracheal system, which is well defined. So there is hemoglobin, all other genes, they are present in tracheal system. So basically, except oxygenation, it has very much same role. And what we do is we, we, we dissected those, we expose this heart in artificial hemolymph in the lab. And then with the high-speed video camera, we can record those beating pattern, if you can see in this beating heart, okay? And you can see much better here. So you can see these beating pattern, you can record them. But then we have a software, we can cut one pixel of this beating heart and draw over time. So when you draw like this, like when we go to our cardiologist, they take our echocardiography, or most of the mouse people also do. We call it mechanical mode or M mode. That's the pattern you can look at, all the diastole and systole. Based upon that, we can determine various different parameters over 20. Some of them are highlighted here. So you can look at the diastolic diameter, systolic diameter, diastolic and systolic interval, heart period, how good this heart is beating, and what kind of irregularity it shows. So we can use these kind of things. Not only that, then you can also look at, in addition to the cardiac physiology, you can look at different developmental stage, lethality and cardiac defect at different developmental stage. And the more frequently, or especially for the screening based study, what we can do, we can inject a dye. So dye will reach to the head through the aorta here, and that means heart is functioning normally. If you have mutant, the dye will not be there. So before doing the cardiac physiology, you can do this setup, basically just a screen-based study if heart had normal function, or it will have some, you know, uh, it will have some blockage or some other kind of defect. You can also look at the lifespan. As you can see, I said they live about 70 days, wild type fly, but some mutant will have half-life. Some will have even, they cannot survive beyond few days. So just a demonstration, we can do that. Of course, we look at cytological level. We can look at these myofibril organization. We can look at the collagen deposition, and we can also look at ECM, like how the ECM or extracellular matrix, all these kind of things. Even we do at, even in this tiny heart, we can look at ultrastructural, like transmission electron microscopy. We can look at this, myofibril. We can look at mitochondrial defect. So I'm just talking about the tool we can use. Then, of course, we have shown that we can do the transcriptomics. So we can do bunch of things that, like most of the mammalian people will do in their heart physiology or cytology, biochemistry, you can call it. I also use a other model in the lab for skeletal muscles, like our human skeletal muscle, Drosophila skeletal muscle, called indirect flight muscle. So this is the very powerful muscle because they have to fly. So if you look at molecular level, they are highly similar. If I show the EM, they will look exactly same. So that means it's a great model for doing human skeletal muscle disease. So what we do is, when there is a defect, these are the fly, they fly always, but if they have a severe defect, they have a erect wing. So very simple thing, like screen-based study. If you have erect wing, now you can do a genetic screening, or you can do a drug screening that can, fix this wing phenotype. And then you can look at more detailed muscle defect. Then you can look at a functional defect. Like these are great undergraduate projects. We have a plexiglass box. They can fly in different direction. Some of them will not fly. Based upon that, we can determine their flight index, how good they can fly. And then we can have other, like we are climbing, like climbing assay, we call it. So we can do these kind of thing also, how quickly in a certain time period they can climb. Of course, we can do cytological study, and you can do the ultrastructural study on the skeletal muscle, as I said, in the heart muscle. Most recently, uh, of course, fly have a well-established brain also. I'm not gonna go too much about that. Just I want to highlight one really important thing. So this is the fly eyes. It's a picture of the fly eyes, which is closely connected to the brain. And some mutant, there is a tau mutant, some of you may know, mutation of tau responsible for Alzheimer's disease. So what you see in a mutant one, you can see eye degeneration. Okay, when we did the same experiment in the presence of circadian disruption, so the phenotype even get worse. So we want to show that, okay, this is when you express, Alzheimer causing protein, it lead to neurodegeneration, which is further, aggravated on circadian rhythm disruption. The other thing we also do lot of activity monitoring, we call sleep analysis, or we can do like what basically it work. It's like a security camera in your home. Basically, you put them in a little vial, and there is a infrared beam that is sensing whenever they move around that tube, so you can sense them and detect their, how active they are during daytime, how active, how many hours they are sleeping, or minute they are sleeping. So we can look at these kind of sleep and activity. Okay, so these are the very basic idea, like cardiac, muscle, to all the way to the some neurodegeneration and circadian activity that my lab does. So I want to talk to you some of my lab project. So my lab has like 3, 4 main project. One of the project is how time restricted feeding and circadian clock, mediated lifestyle can prevent cardiometabolic disease. Okay, so the basic idea is, this is one of my R1 right now I have. So if you maintain this feeding fasting rhythm, it reduces body weight, and it also improves sleep, and it also increases metabolic efficiency. Uh, uh, all these are factor for improving cardiometabolic health. Okay, there is a other project, completely different direction. It's a genetic mutation. The mutation is the gene called LMNA, lemon. So this gene, there is several mutation associated with that. That's the one of the highest disease causing gene known. So that lead to cardiomyopathy, that lead to muscular dystrophy, that lead to a very rare but fatal disease called progeria. So we make that model also. Okay, and then why this project is always very close, because that's where I started my independent career. So just look at Huntington disease. People study Huntington disease for many years, decades. But what I want to look at, it Huntington have any role in the heart? When we express that Huntington in the heart, you can see those puncta aggregate in the heart also, and that lead to cardiac defect. Now we are also looking in the neurological aspect, not just in Huntington. I use a term called aggregation-based cardiac amyloidosis and neuropathy, and then we are looking what is the role of circadian clock on that, and then if TRF can be, can be interventional for this aggregation-based disease also. The last but not least, only started in last 2.5 years, link between insomnia, sleep disruption with the cardiovascular disease. So that include human genetics, that use a Drosophila model. It's a multi-PI grant that also uh, recently funded. And then we have a pending program project grant also. So that also, there is no way I can talk about all these projects. So each of them has one lecture. But what I want to do is I want to cover a little bit of this, the metabolic project and then a genetic project. So just quickly, these are the people who contributed or contributing on this project. So as you know, circadian clock, a 24-hour cycle, and what it does, it prepare us for environmental change. It present everywhere, plant, animal, fungi, cyanobacteria, sorry, and then it's pervasive and cell autonomous. And you can change your circadian clock multiple way. Temperature is one thing, food is other thing, light and dark cycle, well known. Okay, you can change that one. If you look at that, so I don't know where we are. Yeah, so this is like that's where we are right now. So all our day and nighttime activity are mediated by circadian clock. So why we get sleep in nighttime? Because when it's dark, melatonin secretion will go up, and that will reduce your body temperature. That's why you get a sleep. And if you have light, the melatonin, will not go, like down, and you cannot get sleep. So whenever you are sleeping, you should not turn on your light, basically. This is one example, how we have different body activity. Most of them are circadian related. If we look at molecular level, there are 4 different genes in mammalian system. And so there is a transcription and translation feedback. That's how itself a huge lecture, so I'm not going to talk about. I just want to tell that they are highly conserved in fly and mammalian model. So what happened? Our body has multiple clock. Actually, every single, not just a tissue, every cell also have its own clock. And basically, they very much maintain our physiological activity, but there are some behavior that can, disrupt your circadian clock. One typical example is jet lag, and shift to occur, aging, erratic lifestyle. What happens when we have this, this disruption in circadian clock? And you will very much affected everything: sleep quality, depression, cardiovascular, muscle function, you can name it. So it very much affect everything. But the good news is there are some behavior that we think we can fix the circadian clock, and that can promote healthy span. So one of these factors that we are working is time restricted feeding by maintaining a rhythmic lifestyle, and then healthy exposure during daytime. That's also important. Okay. So why this is important? So this is not my study. This is done from like 16-year follow-up study just on human being. What is their breakfast time? What time they are eating? Dinner, and how late they are eating, or if they are eating regular, irregular. So basically, it's a late night or irregular eater have high risk of heart disease. Okay, after controlling diet. And then there is a other beautiful review came out a few years back. So if you have aberrant eating and sleeping pattern, it has negative effect on your health marker that lead to obesity, lipid profile, insulin resistance. All these are the risk factor for the cardiometabolic disease. So what is the motivation? Why we want to do? Like, what is your hypothesis? Can we restore this metabolic rhythm, and then we can improve cardiac health? So what we have done is, these are the fruit fly, Drosophila. They are on normal diet. We split them into two. One called ALF. They have freedom to eat 24/7. The other called time restricted. So if you can look at more clearly here. So these ALF, they eat food 24/7, or whenever they want to eat. The ALF will only eat food during 12 hours during daytime. During nighttime, they will be in just 1% agar. Only reason they don't get dehydrated. Of course, we use the dark and light cycle because it's circadian control. So we want to make sure that circadian rhythm is maintained. So what we do initially, we put, it's a aging model. We put fly for first 2 weeks under ELF and divide it into ELF and TRF. Then we did the experiment in 3 week, 5 week, and 7 week. When I say 3 week, I'm talking about like mid-age human being. And when I say 7 week, I'm talking about 90 plus year old human being in the fly age, basically. Okay. So then we did the experiment at various stage, and we want to check what is their feeding behavior. As I said, the ELF has freedom to eat anytime. Yes, indeed, they eat about one third of, if not more, of their food during nighttime. But these TRF flies, they eat same amount of food, if not more, in that 12-hour period that we feed them. Then you look at the body weight. As you can see, weight increase, then it decrease, and then triglyceride content is also decreased. We also look at, their activity. Previously, I was talking about, like, we put them in, like, in a security camera, and they can move. We can detect their movement and then determine how many times they are, going through that. So what is their activity? As you can see, these during daytime, TRF has more activity, and less during nighttime, but overall activity was not changed, which means we are not counting the exercise as a factor here at least. And then we are looking the sleep. They sleep very well during nighttime, and overall sleep under TRF is also, time restricted feeding is also improved. This is just a pattern how the sleep looks like. As you can see, they sleep way more during nighttime under TRF. So Then we want to what we have done previously. We look at the triglyceride content is down. We look at they sleep better, and their body weight reduced. We want to look at their cardiac health. So if you look at the middle age one under ELF and TRF, you can see that they have a rhythmic pattern. As you can see, the beating pattern of the heart. But look at what happened to this heart in like next 2 weeks. If you compare this with this, you can just tell that you don't have to be any expert just by looking beating pattern. You can look at a lot of dysarrhythmia. But this dysarrhythmia was significantly suppressed when we put the supply under TRF. Okay, as you can also see through M mode at 3 weeks, there is no different, and 5 weeks, lot of dysarrhythmia under ELF was significantly reduced under TRF. Okay, so we did that, and then we want to know, okay, what is the mechanism, how that work? You have to do when you are working in the fly, you have to provide all that. As I said, just showing a phenotype is not sufficient. So what we did is we did the uh, sequencing RNA transcriptomics on the head because we have improved sleep. We did the whole body because body weight was reduced under TRF, and we did in the heart specific also. Okay, at various stage. And when we say in uh, the RNA sequencing in circadian field, you have to do a temporal. You cannot just do like a triplicate or something, as you say, like here in every 3 hour or every 6 hour. Depends upon what you want to show. So what I want to do is I'm not going to go too much detail. So I just want to highlight what we are showing here, a ratio of TRF to ELF. And what happened just in heart, so there is a specific chaperone called TCP chaperone. They are only upregulated in the heart, but in other body at different age, they are either up or down. Okay, they were only upregulated all the time we reported they are upregulated in the heart. And not only that, that's what in the beginning I was saying. So we use all those 7 subunit, and we have mutant version of these available. So we check that if if we impose TRF on those mutant version, they lost the TRF mediated benefit. That means they are certainly involved with this, time restricted feeding is important for delaying aging. So you can see in the Y type under TRF, arrhythmia index is reduced, but not with the mutant. Just one example here. The other unique thing came out. These are 23 component of mitochondrial ETC. Most of you are aware of this one. So they were down regulated in the heart. Okay. So, and then we tested few of them, and at least few of them we tested, and they reduce, actually they delay cardiac aging. So this is not surprising. There, if you look at the literature, there has been, if you knock down even using some drug to certain extent, keep in mind that's really important, because if you knock them down completely, they show severe cardiac phenotype or other dysfunction also. But you slightly inhibit them, they has been like they delay aging in other model also, so that also we show here. Then we also want to know if circadian clock is involved. Okay, so what we have here, you are looking is, this is wild type, you can look at uh, under TRF, arrhythmia indexes went down. But when we took a different version of those circadian clock, cycle, period, time, these are the four circadian clock. So except this period, all of them either they get worse under TRF. Basically, the mutation of circadian clock lost TRF mediated benefit. Okay, so that we shown. But so that was on aging. So now the question is, as you know, obesity and obesity associated disorder, there are too many. I'm not sure I put everything here, just I want to say a few of the things here. Cardiovascular risk are significantly increased, skeletal muscle, these also lead to lipotoxicity, ROS, oxidative stress, you can name it. So what we want to test if TRF will work against this obesity-induced cardiac or skeletal muscle dysfunction. Okay. So what we did is, as I said, we have this skeletal muscle model. So, and that I already shown here. So what I want to do here, so we have this regular diet, and then regular diet, then we have a high fat diet, and then we have a genetic mutant. It's called SK2 sphingosine kinase mutant. So first we are looking is first you want to show whether they are obese or not. If you look at this, the wild type versus any of those mutant under the ALF, they have higher body weight. They are obese fly actually. And then under time restricted feeding, they reduce the weight of the fly, as you can see in both obesity model. And triglyceride content is higher, and that was significantly reduced near wild type level basically. And we also look at the insulin resistance. There are multiple ways you can look at this. So one of this really easy marker called NLEDGE in human or in mammalian, it's called apolipoprotein D. The level of that expression of that has been used a marker for the insulin resistance. As you can see, both obesity models show insulin resistance, which is suppressed under TRF. Then we did the other one. We look at the hemolymph glucose level, which was elevated, and that also reduced under TRF. The other thing we also look at is, now this is the muscle cryosectioning, and then stain with the phyllodin, and red one is the Nile red, which detect the lipid droplet. So as you can see, both obesity model have higher lipid droplet, and they are significantly reduced under time restricted feeding. Okay. So why this is important? If you look at the literature, the people, human being, those are obese and they have sedentary life, and you can see those, the lipid accumulation in the muscles also. That is a lipotoxicity model also. So we did that. Of course, heart function was also reduced. Then we also even look at EM level in the muscle. You can see mitochondrial disarray and some myofibril disarray, and that was also improved with the time restricted feeding. Then beyond that, the muscle, we look at the other adult fat body. These are like, again, you are looking the fat body staining in the fat bodies, which is equivalent to the vertebrate liver. As you can see, they are significantly increased, but lower with the TRF. They are higher in obese model, but they all suppress under time restricted feeding. So then we want to disrupt the circadian clock and see what that happened. And we did that. So we are doing very much same kind of ELF and TRF that I previously talked about. But now we are keeping light on. That means if you keep light on 24/7, your circadian rhythm will disrupt. Okay. And again, we want to see if TRF work on that model. So we did, if you look at first thing I want to say is, when you look this TRF, when you disrupt the circadian rhythm compared to this, if you look at this per se this, so under circadian rhythm, the muscle function is decreased, which is improved under time restricted feeding. Okay, as you can see in both male and female, different age. I'm looking. This is the Y type model, but then we even it was some extent in one of our obese model, sphingosine kinase mutation that I was talking about. Okay, and then we also look at the lipid accumulation. So Y type under circadian disruption, more lipid accumulation, which was also suppressed under time restricted feeding. And of course, the insulin resistance was also down. Okay, if I want to just summarize what I just said, so if you use high fat diet, age, clock disruption, and genetic mutation, they all compromise skeletal muscle function. And then what time restricted, it improved muscle metabolism, muscle structure was better, muscle function, and overall muscle health was improved. Okay. So right now we also did transcriptomics in the skeletal muscle. So this is the model, and we did, and then we are doing the genetic validation. What we have found is something, some pathway, they are upregulated in both obesity model, but some pathway are only specific to diet-induced obesity model, and some are very specific to genetic-induced obesity model. So this paper is we just finalizing. I hope my graduate students are listening to me. We are planning to submit this month. Basically, it's very much ready to go. Okay, if I want to summarize this part of the project, what time-restricted feeding does is improve sleep, body weight, and triglyceride, and it reduced age-associated cardiac dysfunction. Then we also did the steroid muscle, both cardiac and skeletal muscle, that linked with obesity, and time-restricted feeding suppress that, and by reducing fat, fat filtration and insulin sensitivity, by enhancing insulin sensitivity. That means it's reducing insulin resistance. Okay, and there is synchrony between feeding fasting cycle and circadian clock that uh, drive that anabolic and catabolic process. And the last but not least, when you eat, maybe just important what you eat. So that I always say. Let's see how much time I have. Oh, I still have enough time. So whatever I said in the fly model, most of you work in the vertebrate model. So I just want to highlight some of the work that done in the vertebrate model or even in the pilot-based human study we had. So what I'm trying to show here, this is a obese mite. They feed with a high calorie diet and kept them, if they are obese mite, you feed them even high calorie diet. Under ELF, they will have really fast morbidity and they become more obese. But here, under time-restricted feeding, some of that work is not even mine. It's from my collaborator lab, most of it from and some other lab, and they remain obese but fit mice. They are more active than this one. That means my fit mice. And if they feed these lean mice that high-calorie diet and soda, and put them under ELF, they become obese. But time-restricted feeding, they remain lean and fit. So we try to impose that in human being. So what we did is, generally we over the weekend we try to be more relaxed, and only did this feeding during this time restricted feeding during weekdays, not during weekend. Okay, so but there is still benefit when you do for very short time period. We have also shown that in that science paper, if you uh, introduce time restricted feeding very late, still it will have cardiac benefit. If you start for certain time period, stop it, still will have some cardiac benefit. So we have shown that. So summarizing everything, if we call it, so what it does, time restricted feeding does, it reduce blood cholesterol, fasting blood sugar, body weight, body fat, inflammation, and all these factors you can look at, they are improve, sleep improve, endurance also increase. So this is a paper from our own, UAB Dr. Peterson lab. So what she is doing, the early time restricted feeding, she using those human beings. And as you can see, as I said in the fly model, they also show insulin sensitivity is improved under time restricted feeding, beta cell function improve, and this all that insulin is reduced, blood pressure reduced, oxidative stress and appetite, all these functions are improved. So that also has been shown. And more recently, this is again paper from my collaborator. Now they are only enrolled people with the metabolic syndrome. So these are the criteria for people having metabolic syndrome. And under time restricted feeding, most of these factors are actually improved in a small pilot based study that just published late to 2020. Okay, so that's how the time restricted feeding is. So now you are thinking what I'm doing, what we are doing right now in the lab. So what I have done so far, role of time restricted feeding in aging model, then metabolic like obesity model, And now we are modeling a lipid storage myopathy. Okay, so what is this one? Uh, this lead to muscle dysfunction, as name says, lipid storage myopathy, and then it can have a cardiomyopathy, and sometime in some case multi-system dysfunction and accumulation of fat in these tissue. Okay, that's why it's called lipid storage, and it actually caused by a mutation in this electron transfer flavoprotein dehydrogenase, and this is essential for that, as you can see, mitochondrial flavin 1. And there is a very similar disease, but the gene is different. It's called neutral lipid storage myopathy, which is caused by the phenotype is very much same. It can have fatty liver, cardiomyopathy. It also lead to abnormal accumulation of fat in these tissue. But this was caused by most of you know well-known gene ATGA. Okay, so these are the one we are modeling in the lab right now. So what I did is uh, so these have different kind of mutations. Some of them will have point mutations, some will have them have frame shift mutation, some of them will have deletion mutation. So what we did is we just got a genetic mutation of these flies. So this is equivalent to the first one, and that is equivalent to the second one in the fly gene. So I only wrote that. So what I want to do, if they actually show kind of obesity. They have high body weight compared to control, and their triglyceride content is high, glucose is also high, and they just mutation. It's not like a disease causing mutation, a genetic disruption of the gene. That's all I'm showing here. And then it also lead to muscle dysfunction and cardiac dysfunction. So both in cardiac and skeletal muscle. And then we also look at the fat accumulation in the muscle that I was talking about. You can look at that also. So uh, what we are doing right now, we are generating CRISPR based mutation. One of the graduate student projects, she's working on that. And right now we are making some mutation through a biotech company, but we are setting up a platform in the lab. My goal to generate like 50 plus mutation by end of this year using this CRISPR based uh, mutation. Okay. So I just want to switch the gear for like last 15, 20 minutes, whatever I have on this genetic mutation of nuclear lamin. It's called LMNA. This was done by two graduate students. They are already there. I wish they come here too, like the other two came, but they have their own life and she's so nonetheless they are. I wish I really wish they could have come here. They are like wonderful people. They contribute significantly on this project. So what I want to just tell you is this blue one is a inner layer of the nucleus is actually lamin, okay, LMNA. And what it does, as you can see, it has several roles inside the nucleus. It interacts with the chromatin, and it also, a sequestration, a transcription factor. It had lot of cytoskeleton connection through this SWN protein, and then it also involved in signaling this. So basically, nuclear lamin is a building block of nuclear architecture. The laminopathy. So this is a gene consists of about 600 amino acid and over 400 mutated that lead to a different kind of muscular dystrophy. As you can see, they lead to cardiomyopathy with the conduction defect. And then they can have, some cases, they can have congenital muscular dystrophy also. And the other one called lipodystrophy, they cannot process that fat. So that's why, in fact, we can see that fat accumulation in the muscle, but not in the body where it should be. It's called lipodystrophy model. And the last but not least is this very rare and fatal one. It's called progeria. So these kids cannot go beyond their teenager age. And what will happen when you mutate that, the nucleus is collapsed. Okay. So what we did is we make some mutation in the fly model. So as you can see, this the gene is had three major domain, head, rod, and tail. And we use some mutation in the head region, few in the rod region, and few in the tail region. And the region we took these, they are conserved fly, in the fly version of the lamin called lamin C. In human, it called LMNA. And these mutant are linked with the striatal muscle pathology in human. And for some of them, you have muscle biopsy sample is also available. Okay, so first we did just I want to show one example here. So we use one mutation in the IG4 domain or tail domain, and then other one in the rod domain. But here is the IG4 domain. You can look at this is Y type, and this is the one of the mutant, and you can see that there is a lot of dysarrhythmia. And if we look at in more detail, actually, so this is the wild type. You can see a nice rhythmic pattern in the wild type lamin also. But one of the mutant that is in the rod domain lead to dilated cardiomyopathy. If you look at those diastolic diameter compared to control, they are way bigger, and systolic diameter is also bigger. But the other mutant in the rod domain, so that was actually narrow, that like restricted phenotype. So we can differentiate between the dilated and restricted phenotype. Okay. Then we look at the cardiac defect. They can have as you can see, both Y type doesn't have a major defect, but the mutant have severe defect, like they will have defective ostia, they will have non-beating segment, and some of the heart will not beat at all. And then we
Hey,
look at,
Girish.
yes,
Yep. Sorry. Sorry if I missed this. My internet crept out. Can you actually do tachycardia and bradycardia in flies? Is that what we're seeing where those
yes,
stars
yes, you can do
are?
that. So this is like we call it, sometimes it's way higher, we call it arrhythmia, but we can detect, yes, the we can do those kind of things. We have shown that in other models. Yes, yeah, but that's good question. So we can do these kind of thing. And of course, lifespan, as you can see, one mutant lead to severely reduced lifespan compared to control. The other has, even little bit less significant, but still significantly reduced compared to control. So if you look at those heart structure, these are like a circumferential fiber. They actually beat. You can see nice pattern, but here is a dilated one. You can already see a myofibril disarray, and more severe, this is a restricted, the narrow heart. You can see that one also. And what it really came out, what I told you, this lamin is in the inner layer of the nucleus, but in mutant, you can see first of the nuclear are deformed. Their size is not regular like in the wild type. And this, you can see lamin in the cytoplasm. We call it cytoplasmic aggregation of lamin in both mutant we found. So not only just lamin, there is a other nuclear protein called amyrin. Some of you know. So you can see that kind of even amyrin was in the cytoplasmic. So we can so blabbing of nucleus, cytoplasmic aggregation, and disruption of myofibril in both model. Then we want to know what is the mechanism, how that all occurring. So we look at the uh, all those aggregate. So we want to look no aggregate that I was showing in the previous slide. So we want to look at if autophagy is upregulated. So this is one of the marker P62 that you can see in both of mutant. It was upregulated in Drosophila heart. But then we have for same mutation we have human muscle biopsy, and we look at the P62 level is significantly upregulated in same mutation. The number are different, but mutation is same. I just want to highlight that. Then other thing we look at nuclear mislocalization of NRF2. Most of you, some of you are working with this NRF2, so it is in cytoplasm, but what we see in mutant, it actually enter in the nucleus to some extent. And same happen with the patient muscle biopsy sample also. Okay. So then we can do in the fly, we can do a lot of genetic manipulation. So autophagy was defected. So what we did is we use ATG, it's a master regulator of autophagy. Okay. So we use the overexpress that, then we inhibit the NRF2. So we did them individually, and then we did together. In the same ply, we are expressing mutant, and we are overexpressing that and inhibiting this NRF2. So if you look at the arrhythmia index of the mutant, which was significantly suppressed when you use overexpression of the autophagy 1 and inhibition of NRF2. But if you use the mutant version of the ATG1, then it doesn't work. So a functional autophagy is required. And we did the same thing in the wild type background also. And if you look at the other cardiac function is improved, and that was not happen when we have mutant version. And again, in the wild type also you can see. If we look at here, those are those cytoplasmic aggregation, myofibril dish array. This was significantly improved, but not with the mutant version. So everything is not shown here. Physiological function is improved. Nuclear morphology improved. This localization was also myofibril, and viability was also improved. So we did that. So if you want to understand how this is all happening, so these are nuclear envelope protein aggregate, and it lead to upregulation of P62, and P62 is actually binding with the KEAP1. Okay, KEAP1 is supposed to be binding with the this NRF2 in fly, it called CNCC. So now this NRF2 cannot bind with the KEAP1 because KEAP1 is non-specifically binding with the P62. So NRF2 is now released in the cytoplasm where it binds with this detoxy gene, and that lead to mitochondrial defect, all that one. And then we have even shown if we, overexpress ATG, so we can activate that and remove all that one. And even in the downstream one, we can see it lead to inhibition of AMPK and that all called cellular and metabolic stress and cardiomyopathy. Okay, now we are looking the other additional mutant, but this time now in the skeletal muscle here. Okay, so what we see here, these are the different ages, wild type control, then different mutations. Some of the mutant doesn't show any defect to start with, but with age they start showing. But some of them are non-flyer to start with. They are barely flying, and then they become completely flightless. That's what I was referring to you previously. So these are the wing. When they cannot fly, you can see their wing are upheld. Okay, that was previously I was saying. This is held up wing phenotype, correlate with the muscle dysfunction, and it's a easily visualized marker that can be used for high throughput genetic and drug screening. That's what we are doing actually. And of course, it leads to mitochondrial and autophagic defect in the muscle, as we saw in the heart one. Then we also look at the cytoplasmic aggregate, and they correlate more aggregate, more muscle dysfunction that we have shown also. Then what we did for the same mutation, same patient, we got the transcriptomics of human muscle biopsy sample. Okay, and what we found is gene involved with glutathione metabolism was upregulated. Gene associated with the TAUR pathway was also upregulated. Gene linked with this proteostasis or aging was downregulated, and AMPK and downstream signaling of AMPK was also downregulated. So we want to know what is the functional consequence of those downregulation of these gene or upregulation of that. So that's what in the beginning I was trying to show. So we can get a transcriptomics data, but we want to genetically validate that and show the mechanism. And then at the end, our goal is if that can be used for a small molecule therapy. One of the example I want to show you here. So these are nuclear envelope protein cytoplasmic aggregate that lead to upregulation of P62 that reduce autophagy. And what we did is that transcriptomics data show that the AMPK signaling, the transcript was down, SHIRT was down, PGC1 alpha was down. But when we overexpress this AMPK, we can rescue that phenotype. So we tested these one. We can overexpress FOXO, we can rescue that. We can overexpress 4EBP, we can rescue that. But here the TOR was upregulated and the downstream of the TOR S6 kinase was also upregulated. When we use a dominant negative version of this kinase, we can also rescue. Here is one example. If you look at this mutant, it was flightless, but when we use AMPK overexpression, you can rescue their flight phenotype. But when you use AMPK knockdown, it doesn't work. And we use a a GFP knockdown also, just to confirm that it doesn't work either. And then we also look at this child of wing phenotype that was rescue, but not with the AMPK knockdown. Again, the mutant those cytoplasmic aggregate that was rescued with AMPK overexpression to the nuclear structure was not completely rescued, but of course, AMPK knockdown make it worse. And we just also use a ICAR that activate AMPK that can partially suppress this pathology also. So if I want to summarize that section also, this has highest number of disease-causing mutation, but how that work? It's a very complex protein to work with. So we are trying to understand, making a model, and then we show a cardiac dysfunction and aging. Then we look at the cytoplasmic mislocalization of laminin, upregulation of P62, and impairment of cellular redox homeostasis. Then the transcriptomics data, I said that misregulation of AMPK, autophagy, and TOR pathway, and then we modulate the gene, and then we think it can be used for like a potential therapeutic drug target. So just testing those hypotheses. I think this is my last slide, but very, very important to acknowledge all my lab people, my collaborator on different projects, of course, funding. And my lab have open position for graduate student, postdoc, and other researcher. Please contact me, anyone is looking for, and I will happy to take any question. Just I want to show a lot of people here work in cancer. There is a Drosophila model about cancer. A lot of, they say it's a great model, all that. I don't know anything about that. We will happy to talk about that if someone is interested. And then there is other, like additional, if you look at the compare for the obesity or any other model, so these are very much present in the fly model, whatever you are looking for, if obesity or other disease in human, it can be used that model also. I'll just stop here. Thank you.
Well, thank you so much, Dr. Melkani. That was fantastic. Sorry, I dropped out there with internet issues. So I will gladly ask the first question while I'm waiting for other people to unmute. I don't see any in the chat section. You're definitely spectacular at selling the fly model for doing this amazing research. I find the echocardiography on the fly heart truly impressive. My question for you has to do with some of the genetics you were talking about, because flies, if I'm not mistaken, have a slightly different chromosomal structure than eukaryotes, than mammals. How do you deal with like polychrome and stuff like that and getting to some of the gene expression issues, or do you?
See, I don't think that is, I thought that should be a more simpler than that, because it's a very simple 3-4 chromosome. Fourth one is very small. So that is, in terms of genetics, that's never been a problem. That has been the most powerful thing in fly, using that, genetics. But I don't recall anything like that, seeing that kind of issue with their genetics.
Okay, thank you.
Girish, can I ask a question?
Yes, please.
Two things. Can you comment on whether these models are amenable to looking at hypoxia responsiveness or ischemia? Because a lot of diseases and things that a lot of people are interested in obviously have that aspect in terms of energetics and redox and cardiac function?
So that is great question, and lot of that has been done. I don't do it in the lab. I has been used for acute hypoxia model or other hypoxia model. There is this HAP1 alpha that is highly conserved in the fly, and that's like oxygen sensing gene, like hypoxia one that has been well studied in fly model. And yeah, so it's a great model for doing this hypoxia, but I haven't done, but it can be done if needed for any purpose. There are multiple paper on, even they have looked at the gene expression under hypoxia and normexia. So all that has been done by many group, at least 2, 3 group, one in UCSD, one in Sanford Burnham, where I did my part of my postdoc. Actually, I was official in SDSU, but that's where I learned all this heart model in Sanford Burnham with Dr. Bodmer and Karen Ocker group.
Hey, Girish, this is Upender. Great talk. Outstanding. You are adding entirely a new dimension to the pathology expertise. So using Drosophila, perfect model because of this genome homology between the Drosophila and humans. But what are the challenges in translating the findings, what you found in Drosophila to human setting?
Yeah, I mean, challenges are definitely there. In the beginning, when the heart model, if you look at the heart model, only 4 or 5 labs in the world use that Drosophila heart model. And in the beginning, people are not 10 years back when I was doing like postdoc, they said, that's not called heart model, they call dorsal tube at that time. I said, no, it's a chamber structure, it has all. And then people say, what about the other network? Like, does it have any kind of vasculature? And then we said, if you look at those genes, they are present in that model. So in the beginning, it was hard to accept. But then when we did, like people did transcriptomics, including my lab, we did the proteomics, all those human genes, you can see that. And the kind of, manipulation you can that, people start accepting that. And at the end, in my grant or even paper, they'll say, great study, but that's it. Drosophila model. I still hear that, and we address them. Say, take advantage also. Just think about how I am planning to look at like 50 plus CRISPR mutation this year, making in the lab and study them. So, the kind of advantage is, of course, it's not a mouse model, and there is some limitation. It's not 4-chambered heart, but the big, the information you can get from that basic model is very, very informative. And then you can translate that into translational model. Like, out of those 50 genes, like in the laminopathy, Dr. Raj Shekharan is already start making this laminopathy mouse model. So we talk about that. You can look at that, and then we have human muscle biopsy. As you saw, some of the things just mimic what we see in the human muscle biopsy, we see in the Drosophila heart and muscle model also. So it has I will say it's not like, I'm not saying, okay, with this fly model, I'm ready to test the drug, but it's provide very basic information and really quick time and a lot of information.
Absolutely.
Thank you.
Thank you.
Looks like we have one more question. You
Sure.
want to unmute?
Yeah, hi, Girish, this is Maitre. Fabulous talk. I was just curious about in both your circadian studies and your obesity studies, are you looking at the effects and the relationship with the immune system? Does the fly lend itself to those studies? I know they have an immune system. I wasn't sure how much those studies are integrated.
Yes, there is immunity other model that has been studied really well. Even see, if you can see in this slide, there is something they say they have those hemocytes.
Okay. Got it.
So I look at I haven't paid too much attention. Couple of other people also brought that in my attention, and in future, I'm really interested in looking that one too. How time restricted feeding does to immune system. Actually, what we are doing, we have some preliminary data we are about to publish and get more sequencing. We are looking those microbial, those population, what happened under time restricted feeding. Then we have the other model, what happened to stem cell model also, and that especially in the microbial population immunity there. So we're gonna address that in near future. Definitely you're gonna hear maybe later this year or next year.
I see. Thank you.
Yeah.
Well, just to remind everybody, Dr. Melkani is now on campus and part of the pathology family, so please continue the conversation. I know he's eager to build new collaborations with as many of us as he can. So thank you all for joining. Next week, we change gears again. We're having a visiting speaker from the University of Miami, Dr. Montgomery, who's going to tell us about her favorite gastric lesions. So get back to the clinical side as we've been alternating. So thank you all and enjoy the rest of your week. Thank you, Dr. Melkani.
Thank you very much. Have a nice day. Bye-bye.